is thrown into folds. What are these folds known as? Do we know what they're called? These finger-like projections.
Brunner's glands?
What?
The Brunner's glands?
No, you're a little ahead of things. These aren't glands. The villi. The villi. Okay? This increases surface area for absorption. There are three modifications I mentioned. One you can see with the naked eye, and they're visible on the model, those circular folds in the intestinal wall. What are those called? Plicae circularis. Okay? These are microscopic folds that increase surface area for absorption. Those are the villi. Then you got a third adaptation, which you really can't see with a light microscope. It'll really take an electron microscope to see them clearly. Okay, we don't have any electron microscopes here. Your tuition would be skyrocket. Okay, you think your tuition's bad now? Okay. Anyway, those are called microvilli. And when you look at these villi under the microscope, okay, it almost looks like they're fuzzy or ciliated. They're not. It's just that that's the best we can do for looking at the microvilli. And we got some villi, as you can see. But we can't clearly see the microvilli. They're too small to be seen. So what it does appear as, we got kind of a fuzzy border here, almost like it's ciliated. But in fact, these are folds in the cell membrane of the epithelial cells that cover the villi. And because it looks like the bristles of a fine brush, they're called the brush border. There's some very important enzymes that adhere to the brush border. They complete the digestive process. They're called brush border enzymes. We'll discuss those in the lecture later. Okay? But something you can see very clearly here, okay, in the villi, are these structures. What are these? When in doubt, goblet cells. And these are goblet cells, okay? And again, what do? They secrete mucus. That's all they do. They secrete mucus. They do not move the food, okay? I got some answers about that. They move the food. Or no, I got answers about they filter, okay? They don't filter, okay? They provide the sticky mucus in the respiratory tract, which is part of the filtration system, okay? But anyway, they're producing mucus. And again, they're in the epithelium, unicellular glands that secrete the mucus. Okay? Goblet cells. And then you can see more of the villi. Okay? And these, of course, are the goblet cells. And you can see kind of that fuzzy border there. That is the, we call the brush border.
Is it the brush border?
Brush border. Okay, like a brush. Because that's what they look like. Okay, within each villus then, we have a blood capillary network, and we have a lacteal, which is a lymphatic capillary. This is for absorption of nutrients. Why a lacteal? Anybody remember? We got a lacteal in here. Each villus. Villus, by the way, is singular for villi, the plural form of the word. It's to absorb lipids, particularly fats, because we have a curious habit. We digest our fats, okay? They pass across the walls of the villi here, these epithelial cells, then they get reassembled as fats. Now they're too big to get into the blood capillary network, so they go through the more porous walls of the lymphatic capillaries. They're then carried in the lymph to the blood, and we redigest them in the blood. And that's why after a big heavy meal, particularly one high in fats, your blood is going to look creamy because it's full of fats. And it takes a while to digest them. That's why they say when you have a blood test, what do they usually tell you? Fast beforehand. Whether that blood is going to really strain, it's going to be really heavy with fat. So if they're trying to do, LDL and cholesterol tests, you're going to get a really high reading, abnormally high, if you take it right after you've had a big meal. Okay. Something else I'm going to show you. Okay, again within each villus, as I said, you got a blood capillary network, and you've got a lacteal, and then you've got this connective tissue. What's a connective tissue? Release bliss. What's it known as?
A reeler?
2 words. Lamina propria again. So lamina propria. Okay. Connective tissue. Loose connective tissue deep to the mucus. Or I should say deep to the epithelium in the mucous membrane. Okay, if we drop down here and go to the base of the villi, okay, see far here, you can actually see that there are tiny little pits between adjacent villi. There are tubular pits. Okay, and these are the intestinal crypts or crypts of Leverkusen. You can see it down here. Intestinal crypts or crypts of Leverkusen. And then you got a band of muscle here. Again, it's smooth muscle, like we saw in the stomach. Okay, and this is called the muscularis mucosae, and again, that marks the outer boundary of the mucosa. Deep to that, you're in the submucosa. That's what we see here. This is the submucosa. And it's full of glands. Now, what are the glands? You mentioned it before.
Brunner's glands?
Brunner's glands, also called duodenal glands. What do they secrete?
Enzymes?
Mucus.
Okay, what kind of mucus? That's half an answer.
Alkaline mucus.
Alkaline, yes. Why alkaline mucus? Why do you want a basic secretion going into the small intestine, in the duodenum? What's coming into the small intestine?
Chyme.
Chyme. And what is chyme? An acidic paste, very acidic. The digestive enzymes in the small intestine do not work well, if at all, in an acidic environment. What you'd end up doing is denaturing your enzymes. So you first have to bring that pH up to a workable level, which is usually slightly basic. Okay, neutral or slightly basic. That's the purpose of the secretion of these Brunner's glands, or also called duodenal glands. You can see it. And the mucosa here, the submucosa rather, is just full of these glands. Another way to help neutralize the acidity is secretions, or are secretions, from the pancreas. And these secretions basically are high in bicarbonate ions. Those are stimulated by a hormone, which we'll talk about later in lecture, called secretin. Okay? So again, you ask what these are, Brunner's glands, or you can just simply say duodenal glands, which may be easier to remember, okay, and probably easier to spell. Okay? And again, they're in the submucosa of the duodenum, but only in the duodenum. Okay? Then we get to the muscle layers again, just like we saw before. We have 2 muscle layers. Okay? The inner layer, next to the submucosa, you can see, runs around the circumference of the organ. This is circular smooth muscle. The outer layers we saw before runs parallel to the longitudinal axis. And coming down the plane of the screen here, that is longitudinal smooth muscle. So same thing like we saw before. 2 muscle layers, inner one, adjacent to submucosa, circular smooth muscle. The outer one, longitudinal smooth muscle. Remember that. When we get to the next organ system, it's going to be reversed. But for now, the gospel is inner circular, outer longitudinal smooth muscle. So if you're asked, what's the type of tissue? Smooth muscle.
Smooth muscle.
Arrangement?
Circular.
Or longitudinal. Okay? Don't be stymied. What do you mean by this? Basically giving you a dead giveaway. It says type of tissue arrangement. It's usually smooth muscle. Arrangement is either circular or longitudinal. Okay? Now, the outer layer here, again, is serosa. You see it sort of peeling away here. Simple squamous loose connective tissue. But much of the duodenum is retroperitoneal, which means this is not synonymous with visceral peritoneum. It's just simply the serosa. So if I ask retroperitoneal organs in the GI tract, much of the duodenum, ascending and descending colon, also retroperitoneal. Okay? The other parts are within the peritoneum. All right? Can you think of an accessory organ that's retroperitoneal? Don't say kidney, because that's not part of the digestive system. But that is retroperitoneal.
The gallbladder?
What?
The gallbladder? Huh?
Pancreas.
What did you say?
Pancreas.
Yes, very good. Pancreas. Retroperitoneal. Okay. Thank you. Okay.
Still
Who said it? Okay. You're the mouthpiece, okay? I didn't hear her. Anyway. Yes. Okay. Wouldn't hurt to know those retroperitoneal organs. Okay, let's look now